Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, Model Tumor Host Interaction in Liver Cancer Using Spheroids. I am pleased to welcome Liqin Zhu as your presenter. Dr. Jude is an assistant member at St. Jude Children's Research Hospital. Her research program focuses on studying the role of tumor-liver interaction in the metastasis of pediatric and adult liver cancer. Her work utilizes 3D spirit-based models to spatially place normal and malignant liver cells in their physiologically relevant relationship to study the molecular and cellular changes induced by their interaction. A warm welcome to you, Liqin. Following the presentation, we will have a Q&A session and we will welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Liqin's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. I will now hand over to Liqin to begin their presentation.
Hi, hello, my name is Liqin Zhu. I'm an assistant member in the Department of Pharmacy and Pharmaceutical Sciences at St. Jude Children's Research Hospital. So first of all, I'd like to thank Technology Networks for providing this great platform for us to exchange our latest technical advances in helping us understand human disease. So today I'm going to talk about our effort on modeling tumor-host interaction in liver cancer using spheroids. So I think it's a good, accurate statement that one of the biggest breakthroughs in cancer research in the past 10, 20 years is our increasing appreciation of the importance of non-tumor cells in tumorigenesis. There are many different types of non-malignant cells that surround and feed tumor cells, such as endothelial cells that form the blood vessels, immune cells, myofibroblasts, signaling molecules, and the actin matrix. All these together form a highly dynamic, ever-changing tumor microenvironment that influences and even dictates tumor cell behaviors. What I want to talk to you today is a slightly bigger version of this tumor microenvironment that is present within a tumor mass. That is also very specific to liver cancer, which is what I study. That is the liver. A solid tumor mass apparently doesn't exist by itself. In most cases, it grows within its host organ, and the normal tissue of its host organ provides a very rich peri-tumor microenvironment that incorporates into tumorigenesis and is also being constantly reshaped by tumorigenesis. In fact, liver cancer is one of the best known solid tumors whose development has everything to do with its surrounding liver tissue. More than 90% of HCC, the most common liver cancer in adults, arise in the context of hepatic injury and inflammation. Persistent inflammation is also known to promote and exacerbate malignancy in the liver, as well as its drug resistance, metastasis, relapse. So before I get into detail of our study, I'd like to give you a quick introduction of the cellular organization of the liver. So liver is the largest internal organ of our body. It is relatively homogeneous in terms of its cellular organization. No matter where you cut into liver, you will find this basic liver function unit called hepatic lobule. It is estimated that there are about 1 million hepatic lobules in one liver on average. Hepatic lobule is a hexagon-shaped structure with a central vein in the middle and 6 portal triads distributed around the edge. The main cell type consisting of this hepatic lobule is hepatocytes, the predominant liver parenchymal cell type that take care of the major function of the liver, such as energy production, metabolic and hormonal processes, detoxification, and many others. But there are also a group of relatively less known population in the liver called non-parenchymal cells, including cholangiocytes that form the bile ducts, Kupffer cells, which are the largest resident macrophage of the body, endothelial cells that form the blood vessels, and hepatic stem cells, which we'll talk about more later. These non-parenchymal cells together form the structure support transportation system and take care of immune communication and injury response. So for these cells that are present in the liver, they also, like other organs, can sometimes go wrong and produce malignancies in the liver. So there are three major subtypes of liver cancer. Hepatocellular carcinoma, HCC, we just talked about, it is the dominant liver cancer in adults. And cholangiocarcinoma, which is the second most common liver cancer in adults. And hepatoblastoma, which is the most common primary liver cancer in children. So my lab actually study all three type of liver cancer here, but today we're going to focus on cholangiocarcinoma. Why? Because it is one of the deadliest cancer overall. So it is considered to be originated from cholangiocytes based on its morphology, but there are still many debates over the origin of cholangiocarcinoma. But this cancer is just very hard to treat. It is often diagnosed late, and it has very rapid progression and early relapse. Even the tumor is completely removed by surgery, and it is found to be profoundly drug resistant. So far, there's very few drugs that actually can target the cholangiocarcinoma tumor cells. So overall, the 5-year survival rate of the patients is less than 10%. So this is really a very bad disease that we need to understand why it presents all these difficult properties that resist the treatment. So it is believed so far from all the studies that the highly heterogeneous tumor microenvironment that present within cholangiocarcinoma may contribute a lot to its difficulties, to this aggressive behaviors and the resistance to treatment. So I'd like to give an example about the cellular or tumor microenvironment that I talk about, the heterogeneity of cholangiocarcinoma tumor microenvironment. So this is a co-staining of 4 different markers that show on the screen. So blue is DABI, which stains nuclei. Yellow is PCK-PEN cytokeratin, which stains the endothelial cells, and smooth muscle actin is red, which is a marker for myofibroblasts, and the green is CD45, which is a marker for all hepatopoietic cells. So just only these 4 markers, you can see how a cholangiocarcinoma tumor looks like. So these cells are just organized or disorganized in a way that they have this really magnificent heterogeneity across different regions of the tumor. So, but what I want to bring your attention to today is the liver that next to this monstrous tumor mass. So you may notice that there are actually something wrong with this liver. There are very weird cell organizations that also have these different cell types within. And you might ask, this is how a normal liver looks like? What I can tell you is it's not. This is exactly the same staining that we did on the far tumor liver from the same patient. So exactly the same liver, basically. And you can see how this far tumor liver looks different compared to this near tumor liver. So of course you can argue that, yeah, maybe the patient has some pre-existing condition that make the liver, a certain area of liver abnormal. That's why actually the tumor eventually arises in this region. So of course, this is also always the difficulties of studying patient tumors, because you get a one-time shot. Basically you get sample from the resected tumor, and then this is how it looks, and then it's really hard to tell what leads to what we have seen here. So that's where our research model can come in. So in my lab, we have generated a immune competent metastatic cholangiocarcinoma allograft model. So this model is based on a genetic mouse model that we have reported previously. So this genetic model has the P10, P53, these 2 very important tumor suppressors removed or deleted from a prominent one expressing liver progenitor cell population. So, and the cells, we also have this ZS green, a modified GFP, a green fluorescence protein, introduced into the tumor cells. So the tumor is green. Even to our naked eyes, this is very strong green fluorescent protein. But under fluorescent microscope, we can easily visualize the tumor cells. So what we did with the genetic models, and once we get the tumors, we take them out, culture them in vitro, and get the cell lines. And then we inject these cells back into immune competent mouse strain, so back into their liver. So then what we have is an allograft model that we can really track the tumor development. So as you can see from here, we have a line that actually produces very aggressive cholangiocarcinoma that is already metastatic within the liver and also travel to the lung and form multi-nodule lung metastases. So the good thing about the allograft model is it starts from normal background. So there's no pre-existing condition present in these animals. So as you can see, this is a alpha-smooth muscle immunohistochemistry, which stains the myofibroblasts within the tissue. So this is 1 month after allograft. So you can see the tumor here and the liver surrounding it. And then you may notice that the myofibroblasts are already activated after a month of the tumor genesis. So and the liver still look quite okay, although the tumor is already forming. But then 4 months into the tumor development, then what do you have? Probably can notice that how liver looks different now. So compared to what I've shown previously about this patient tumor, you can see yes, liver is being changed by a tumor that are growing nearby. So this is where everything started. So because we found this liver tumor interaction is extremely fascinating or very exciting, a new area that we need to focus on. So how the liver is responding to tumor genesis and how tumor will also respond to the response that liver produce when the tumor is growing. So this interaction is something that we are really interested in. So there are different research models you can use to study tumor liver interaction. So as I showed you previously, that you can use in vivo animal models because these models, if you do everything right, they can actually recapitulate the after tumor development very well. So the mouse tumors that we generate in our models, some actually look very, very similar to those tumors we've seen from patients. So, but the difficulties of studying, of using an in vivo animal model is that it is very difficult to track and dissect specific cell-cell interaction because, what we have seen in vivo is always a systemic response, and also can be time-consuming. As I showed again, previously said, we'll have to wait 3, 4, 5 months to finally get to a stage that the tumor becomes really aggressive. So this is where the in vitro cellular models come in. Because they're much easier to manipulate, and we can track individual players and to understand their contribution to tumor genesis. But compared to the in vivo models, they are also very different. It's also almost not possible to fully model the systemic tumor response or tumor genesis, is because there's only limited cellular models that we can put into such in vivo models. But if you have these two both together, then eventually, they will help us to have a better understanding, or at least we can try to explain what we have seen in the patient tumors. So so today I'm going to focus on our in vitro cellular models that we use to study tumor liver interaction. And we are most interested in using 3D cellular models because apparently, they can mimic the cellular organization of the liver much better than standard 2D cell culture. So there is this type of 3D model called organoids. Many of you may have heard of them already. So they have been widely used to study normal and malignant development in many different organs. So there are 3-dimensional stem cell cultures that are self-organized into many organs. That's where their name came from, organoids, through cell sorting and especially restricted lineage commitment in a manner similar to in vivo. But there are some difficulties of using organoids to study liver because it is found that organoid models are not very efficient for hepatocytes, likely due to the lack of a designated stem cell population supporting hepatocyte regeneration. So it is actually a very interesting topic that I'm not going to talk about today, but liver is actually exceptionally regenerative. So if you have liver damage, you actually induce hepatocyte regeneration right away. But then still there's a lot of debates like what is the root or the cellular origin for liver regeneration. So there's still not a very fully identified stem cell population within the liver. Nevertheless, organoids model doesn't really work very well for hepatocytes. The plating efficiency is less than 0.1%. So if you play 1,000 hepatocytes into organelles culture condition, you might end up having one organelles growing out of it. So that's not the model we want to go for. So that's where we have this sphere model, which is also a type of 3D cellular model, but it starts from multiple multi-lineage cells. So basically, you just dissociate the liver and get a single cell suspension of hepatocytes, and then if you spin down, and then have the cell aggregate on the bottom, then they will grow or organize into this structure called spheroids. And you can see that they also maintain very well of these different differentiation markers of hepatocytes. So it is very efficient for hepatocytes, although these spheroids are typically short-lived. They don't last over 7 days, and because hepatocytes will start to de-differentiate and start losing their differentiated properties. So you cannot use these spheroids to study hepatocyte-related biology anymore. And it has been found that NPCs, non-parenchymal cells, facilitate hepatocyte spheroid formation, but there are not a lot of studies that are focusing on NPCs in this type of 3D models. Indeed, there are actually no NPC-specific spheroids that have been reported so far. And these type of models mostly used to study liver physiology, not cancer biology. So that's where we decide that, let's try using these spheroid models to see whether we can actually start, just to investigate liver tumor interaction. So this is what we have reported recently in disease models and the mechanisms about how we use spheroid model to study the function of non-parenchymal cells in early liver tumorigenesis. Then Dr. Chen Tian is the lead author of this paper. And basically what he did was using this very specialized multi-well plate called agrawell from stem cell. Because this type of plates actually allow you to generate spheroids very easily. So what Chen did was to get the single cell suspension from the primary, from the liver, from that mouse liver. And we use a mouse strain called MTMG. Basically, this mouse strain has a very strong TD tomato, a red fluorescence protein expressed in every single cell of his body. So when we get the liver cells out, they're all fluorescent for, they all have RFP expressed, so we can easily visualize these liver cells under microscope. So so we have this, we call it WLC, whole liver cells, which means that we did not fractionate the liver. So basically just, we take every single cell that we get from the liver and try to make spheroids out of them. And we also have these HC spheroids, this hepatocyte specific spheroids. And we also have non-parenchymal cells fractionated out of the WLC whole liver cell and then try to get them also form spheroids. So as you can see, by using just 125 cells per microwell, then we can generate WLC spheroids by day 7. So and hepatocytes, just as being reported before, that it's very, very easy to form spheroids. So again, 125 cells per microwell, then you can have this beautiful hepatocyte spheroids forming by day 7. And non-parenchymal cells, indeed, there's a reason why NPC spheroids have not been reported before, because we found, yes, it was quite difficult for them to aggregate and form spheroids. But we found that when we use a larger agrawell and have 2,000 cells put into 1 microwell, then by day 7, they also manage to come together and form spheroids like this. So and then, we did the basic IHC histological characterization of the spheroids that we generated by using different markers for different cell lineages of the liver, such as Kupffer cells, stellate cells, hepatocytes, cholangiocytes. So they all express these markers as we expect them to. And what we found a little bit interesting is that the CK19 expression in the WLC spheroids and HC spheroids, because it is a marker for cholangiocytes. But what we found is very interesting that hepatocytes, which are not supposed to have cholangiocytes, they actually have more CK19 expression than the WLC, which should have cholangiocytes in them. So this is something related to what had been reported before is like hepatocytes. Indeed, they're not that stable in vitro culture. They de-differentiate quite quickly. So then once they de-differentiate, they start to gain the expression of this kind of cholangiocyte marker, including CK19. So indeed, if we scan through all these different spheroids that we have generated, we often can find this type of really weird looking hepatocyte spheroids in our culture. So these spheroids, you can see that have a lot of CK19 expression with them. And these CK19 positive cells are also very, very proliferative, that labeled by Ki67, a cell proliferation marker, compared to those CK19 positive cells in the NPC spheroid, which have a lot of cholangiocytes. They are even more proliferative. So we don't see that in the WLC spheroid culture. So we believe that this is probably an indication of hepatocyte de-differentiation. So then, this actually, because we don't see that, these kind of spheroids in the WLC spheroids, so we believe that it, again, as has been reported before, that hepatocytes are being stabilized by non-parenchymal cells nearby. So this is an interesting thing, because we found, this shows that for a cell to be them, you have to have the right neighborhood around you. So NPC, the presence of NPC cells are very important for the maintenance of the hepatocyte properties in this in vitro system. So then we also did some characterization to show that, yes, these 3D spheroid culture can maintain the hepatocyte property better than the 2D culture. And also they have, a very nice expression of its marker genes such as albumin and CYP3A. And then, now we have this normal spheroids. So we want to study liver tumor interaction. Of course, then we have to have these tumor spheroids. So we found that this PPTR, we call, the prominent one, P10, PP3, rosa, azeous green tumor cells, they form spheroids extremely easy, much, much more efficient than our normal liver cells. So using the same culture condition, 125 cells per microvol, just after 24 to 48 hours, we can generate these beautiful spheroids using this PPTR tumor cells. So these PPTR tumor cells also grow very, very efficiently under the organoid culture condition. But you can see from this picture that, these organoids are all different sizes, different shapes. So, so organoids, be very difficult to use them in any downstream quantitative analysis. So that's why we went for the spheroids, which, in their size, in general, they are quite homogeneous. So what we did is just mix these green tumor spheroids with our red liver spheroids. So we tried this 10-to-1 mixing ratio, so 10 normal, 1 tumor. Because we know that tumor cells grow way better than normal compartment, these normal cells. So, we don't want them to overgrown quickly or shadow over the normal cells. So we don't really we're not able to visualize their interaction that well. So this 10-to-1 ratio that we tested that, we found actually worked pretty well. So from these pictures, you can see how, easily we can tell normal cells and tumor cells apart by their fluorescence. And then we found that once we put them together, these cells start to aggregate. We don't know why, but they like to stay together. So this is suspension culture condition. So all the cells are floating in the media. And we found the hepatocytes aggregate with tumor cells much more, most quickly compared to other conditions. But by 48 hours, then all these cells pretty much all come together in different conditions. So then once they form these co-spheroids, we call them co-spheroids. So and then we monitor the changes in the area of the fluorescence to see how tumor cell expansion can be affected by different cells that are co-cultured with them. So what we found is quite interestingly that tumor cells expand very nicely when they are co-cultured with the hepatocytes. Similar, but a little bit slower than those for those tumor cells that co-culture with WLC spheroids, their expansion rate was slightly lower compared to those in the hepatocyte co-culture condition. And for those that are co-cultured with NPC spheroids, they barely changed in terms of their GAP positive area. So And then, we did the quantification of the GAP area in these co-spheroids over the time, 7 days of co-culture, that you can see that, yes, for those that are co-cultured with NPC, they just not able to grow up that much at all. And then for the WLC-T co-culture condition, they are able to grow to a certain level. And for those that co-culture with hepatocytes, they grow as nicely as those tumor spheroids that are just cultured by themselves. So, and then we want to double check that, that the tumor spheroids grow differently when they are co-cultured with hepatocytes versus non-parenchymal cells. So this time we did the 5:1 mixing ratio of the hepatocyte spheroids and the non-parenchymal cell spheroids, and then just monitor the green fluorescence area. And again, very, very, very obviously that the tumor cells that are mixed with hepatocytes grow very, very quickly. Just, this is after 4 days. You can see that the cold sphere is pretty much all GAP. But then these cells that are tumor cells that are with NPCs, they are just there. I mean, they don't really change much at all. And then it's again quantification. So, and of course, just the GFP area is not just, tumor proliferation, because this is 3D culture and we are basically just taking 2D images. So what we did was taking out these co-spheroids, embed them, section them, and staying for a cell proliferation marker, again Ki67. So and then you can see that indeed these green tumor cells, the co-culture with hepatocytes, definitely are more proliferative than those that are co-culture with NPCs. So and this is the quantification here. So overall, what we concluded from here is that NPCs, not hepatocytes, are able to suppress tumor cell growth. And then, of course, the next question is why, right? Why NPCs are able to do this? Excuse me. So what we did is, we examined the co-spheroid that we generated by mixing the WLC spheroids with tumor spheroids, because WLC spheroids had all these different NPC cell lineages there. So we want to see what cells actually responded to tumor cells interaction. So what do we notice after gone through many different markers? What do we notice? That hepatic stem cells seem to have changed quite a bit. So and the first row column is the just the WLC spheroids by themselves. When they're putting together, they form this gigantic co-spheroids, and then you can see these alpha-smooth muscle actin positive cells. They're just randomly distributed through the co-spheroids, but they have very limited number here, and they're not growing that much at all. And then in the co-spheroids that have WLC and tumor cells, then I hope you notice that there are actually accumulation of these activated hepatic stellate cells that express alpha-smooth muscle actin that exactly sits at this interface, tumor-liver interface. So, and then when we look at the proliferation rate of these tumor cells, those tumor cells that are next to this band of activated hepatic stellate cells indeed grow much less compared to those that don't have hepatic stellate cells around them. So we're excited to see this, because this is what is what happens in vivo in patients as well. So these are two I see ICCs, the intrahepatic cholangiocarcinoma patient tumors. So I hope you can appreciate that oftentimes you will see that there's actually a barrier that formed by alpha-smooth muscle actin positive hepatic stellate cells that sits right at the tumor edge, seeming to be like blocking the tumor genesis from the liver, from invading into the liver. So and this is even most obvious in early stage tumors. So so we're excited that we think our in vitro co-culture model indeed captures some of the tumor liver interaction that are happening in patients. So then I talk about active hepatic stellate cells. What do I mean by that? So a little bit of introduction of this cell population, hepatic stellate cells, HAC. So they're not hematopoietic stem cells that people use to, for this abbreviation HSC. So hepatic static cells are a type of pericyte in the liver. So they sit between the endothelial cells and the hepatocytes. They're considered as quiescent in normal liver. And of course, they're not just sitting there doing nothing. They are actually the largest vitamin A storage cell in the body. But these cells are very, very sensitive to liver damage. So then once some kind of damage, either induced by non-malignant stimuli or just by tumorigenesis, then they will activate, and then they will start to express this mild fibroblast marker of smooth muscle actin. And indeed, in the liver cancer cases, then people have done very sophisticated linear tracing studies, and then found that HSCs, static cells, are the major source of liver cancer associated myofibroblasts. So yeah, so we know now, it looks like the hepatic static cell activation actually slow down the tumorigenesis in our in vitro model. But we are also wondering what do they do to the normal compartment? Why they just sit there right at the border? So what we did is, first of all, we look at the hepatocytes and tumor interaction, because we didn't notice that the green fluorescence in our co-culture system expand very quickly. At the same time, it's the diminishing of the red fluorescence. So tumor cells are expanding and hepatocytes are going away. So that indicates some type of killing of hepatocytes by tumor cells. So we want to visualize that even better. So we put these different type of spheroids on the matched coated plate. So basically, instead in the suspension, we put them relatively in the even plane, so we can visualize the interaction better. So what I want to show you is these day 147 image from the same area, and I hope you notice that how hepatocytes, actually tumor cells actually crave for hepatocytes. They move close to the hepatocytes very quickly, and then just eat them. Basically, I just like to use this word, eat. So you can see like they are just literally swallow this red hepatocyte organoid, spheroids. So in different areas, you would see the disappearance of the red fluorescence, and the expansion of the green fluorescence. But even, those hepatocytes that are not in direct contact with tumor spheroids, you can see they gradually also diminish. So they're just not able to handle tumor cells being around. And then what we did is, so like these two outcomes, the hepatocyte just was like, okay, no, I'm dying. So tumor cells are happy that they are able to have this food that feed them. So and then what we compared was these spheroids we generated by mixing hepatocytes and static cells together. So and then put them together with the tumor spheroid, exactly the same culture condition. And then now what we notice, yes, tumor cells still attack these normal spheroids, move over and then get them surrounded and try to swallow them. But then now you can see like they seem to have some kind of difficulties in swallowing these red spheroids. So over time, yes, red still gradually disappear, but then the rate of the reduction of the GAP, reduction of the RP fluorescence is much slower compared to the previous condition. So so we quantify these, we call it partial and intact red spheroids after 7 days. So partial we define as those spheroids that have the red fluorescence area reduction more than 50%. So they are still there, but they have lost about 50% of their RP area. And for those that are intact spheroids, normal spheroids we define as their area RFP area reduction less than 10%. So for both type of normal spheroids, we can see that they are more in the co-culture condition with the tumor and the mixed hepatocytes and static cell spheroid compared to the hepatocytes only spheroid. So but we want to do this even better, so to visualize the interaction of the killing even more obviously. So Liqin Li, who was the former lab member, who did this 2D co-culture. So I think it's also pretty interesting, very straightforward. So basically, she plates the hepatocytes or a mix of hepatocytes and static cells as 2D sheets on the bottom. And then she sees like minimal number of tumor single cells onto the top. So there are about 20 tumor cells that she sees in each condition. So then you can see the tumor cells quickly start to grow, and then over time, this is like 3-week co-culture, and then we look into the area of the original seeding area and see how much green we will have, how much red we will have left. And then it is quite obvious by week 3, then in the bottom condition where we have static cells, there are a lot more red area compared to the top. And this is the quantification. And we notice that this change or this difference happens typically around week 2. It seems like hepatocytes are trying to hold, but by week 2, they cannot hold anymore. So their fluorescence drops very, very quickly. But when there are static cells around, then this dropping is much more prevented or slowed down. So overall, we think hepatocytes are actually being protected by static cells when there are tumor cells around. So far, what I have shown you is that we think WLC spheroids are a very useful in vitro model to study both the parenchymal and non-parenchymal compartments of the liver, and especially how they interact and help each other bound out, form a team to interact with the tumor cells. And we found the tumor-liver interaction induces static cell activation very quickly at the interface, both in vitro and in vivo. And so far, we found the hepatic static cells play a hepatoprotective role in liver tumor interaction. So there's a small problem here. So what is that? Because hepatic static cells or myofibroblasts have been found, at a very high density in many liver cancer cases. So they have been widely studied over past 10-20 years. So then from all these studies, then the message is quite consistent that these static cells, active static cells or myofibroblasts, they are pro-tumorigenic in liver cancer. So, but what we have shown is that they are actually suppressive, right? They suppress tumor growth, and they are actually protecting hepatocytes. So why is the difference, right? So I want to point out that in the past, all the static cell or myofibroblasts related studies in the liver cancer are studying those intratumoral cells, intratumoral HSCs or intratumoral myofibroblasts. We call them IMFs. So because these are the cells that you can easily collect or easily access to, because when the tumor is resected, then you can get these cells that are mixed within the tumor. But what we are studying here are those peritumoral myofibroblasts or active static cells, the PMFs. So those that are actually coming from the liver and then forming the barrier between tumor and the liver. So and we think just the location difference probably is why their function can be so different. So our hypothesis is that the function of hepatic static cells or myofibroblasts in liver tumorigenesis is spatially defined. So and it's very easy to model this two type of interaction because now we have this tool of spheroids. So we can easily mix fibroblasts within the spheroid to have them coming as IMFs, or we can plate them onto the plate first and then have the spheroids dotting on the top. So then we model this peritumor interaction between these two cell types. So here are some examples that are of the cells or the spheroids that we generated. So we either have the IMFs, the red IMFs mixed within the tumor spheroids, or have the PMFs that are plated around the tumor spheroids. So I want to emphasize that these MFs, myofibroblasts we use are identical cells. All the difference is just we mix them with the tumor cells or we plate them outside the tumor spheroids. So and this study is reporting this recent publication from my lab. In oncogene, and Dr. Chen Tian is again the lead author. So he has put a lot of effort try to understand the spatial temporal regulation of tumor liver interaction, specifically tumor myofibroblast interaction. So the main message in this study is that, yes, just as our 3D suspension co-culture that I have shown before, that when we have the PMFs played outside the tumor spheroids, indeed they have very strong tumor suppressing effect in terms of the tumor growth. So the tumor spheroids expand much, much more slowly when there are fibroblasts played outside of them. And when we mix the fibroblasts within the tumor, they actually promote the spheroid growth. But even inside they're promoting, but still, if you have PMFs, then the sphere expansion is also slowed down. And again, of course, the area of the GAP tumor sphere is not just it's not direct representative of the tumor proliferation. So this first quantified the size of the spheroids by their GAP fluorescence, and also we did the cell tracker proliferation assay, which I'm not going to get into detail, but basically we use a dye to indicate the cell division. And what we are seeing here is that, yes, when we have IMFs mixed within the tumor spheroids, then proliferation goes higher. And then, but in both cases, when you have PMFs played outside, then you can see that the tumor proliferation slows down. So the conclusion is the PMF suppress tumor growth, IMF promote tumor growth. However, there's always a however there. So what we found is if we maintain this culture longer, and something's happening here. So in these conditions that I have PMFs in the culture on the bottom layer, then these tumor spheres become hairy. So they have this processes growing out of them. So and some actually become disseminating tumor cells. So and then we notice that this phenomena is less in the IMF condition, although when you have PMFs outside, also seem to make the tumor cells more invasive. So the overall we found PMFs are able to elicitate tumor cell invasion and dissemination. So this suppressed at the early stage, but then this suppression actually leads cell to the tumor cells become more aggressive. So and once we have this knowledge, then we went back to our initial 3D co-culture. Then yes, it's already there. So if you look closer, actually in the WOCT or MPCT co-culture condition, then I hope you can see that there are actually cell invasion, tumor cell invasion and dissemination already occurring there, which is not that obvious in the HC tumor co-culture. So and then once this happens, what happened, MFs also change because tumor cells going out, then this MFs going in. So then there's a PMF to IM evolution, we call it, or change in vitro in our co-culture system. So this, again, this tumor MF interaction is highly dynamic. So and then this also modeled in vivo that I have shown you this before. So at that time I let you pay attention to the changes in the liver, but then if you look at the myofibroblasts in the tumor in the 1 month allograft, then they're basically around mostly at the tumor edge, tumor border. So, but then at 4 months, you can see that these cells are just pretty much everywhere within the tumor mass. So they have already infiltrated into the tumor very efficiently. So in summary, so what we've shown again in the early phase, we think PMF are being activated by tumorigenesis on tumor border, and they actually control the local growth of tumor expansion. So, but then, gradually then tumor cells actually under the suppressing environment, they become invasive and disseminating. And then at the same time, the PMF will infiltrate into the tumor and become IMFs. And then, and then turn from an anti-tumor player to pro-tumor player. So, and we have other studies that will focus on later stage, even later stage of the metastatic growth, but I'm not going to get into that. So technical wise, what we have shown here is, spheroids, we believe, are a very useful model to tackle tumor spatial omics because you're able to group, your self-interest together and place them in different spatial relationship. So I have shown you, what we have done to compare PMFs versus IMFs. But then, go back to this original image from a patient tumor that, a fibroblast, it's just, there's this very small part of tumorigenesis of cholangiocarcinoma. There are many other type of, tumor microenvironment cells that are within the tumor mass and also outside the tumor mass. So we also, confirm that using our animal models. So we did RNA sequencing of normal liver versus those tumor adjacent liver we collected from our allograft model. And there are a lot of changes happening in the liver, including the reduced liver metabolism and on and off of some cancer development related pathways, and a lot of immune activation going on. So we believe that this model can be further expanded to study other cell types, other malignant cell types that are play a role in tumor genesis. For example, can we compare P tumor-associated macrophages versus I tumor-associated macrophage, or those T cells within and outside the tumor mass, and then study the transcriptome, secretome, and the metabolome. There are a lot of potential applications of this model system. So with that, I'd like to thank all the people that are involved in this project, especially our Dr. Chen Tian, who have contributed like many hours of his work, just to try to understand liver tumor interaction, and then people that are in my lab and involved in this work, our collaborator, especially Dr. Glazer from the University of Tennessee, who have helped us achieve these access patient tumors, and our folks in the St. Jude Computational Biology Department, and our funding source. With that, I'd like to stop here and open the floor for questions. Thank you very much for your attention.
Thank you very much, Liqin. As with all the talks, we are likely going to have lots of questions from the audience. I can see we have received some questions already, so I'll move straight on to the Q&A now. For the first question, how does the knowledge gathered from your model system help us understand liver cancer biology?
Yeah, so thanks for the question. So I think, the best part of this in our study is that we broaden our view, about liver cancer development from just the tumor mass into a bigger picture of, involving the host organ into in this interaction. So in the past, because of the limitation of the sample, you basically get the tumor tissue, then what do you study is the tumor tissue. But then we all know that a cancer development is a systemic process. So there are a lot of involvements, response, defense from the surrounding tissue. So I think our model, the most unique message here is that yes, it's not, tumor is not just about tumor cells. There are a lot of signals, interaction that happening between the tumor and its host organ.
Thank you, Liqin. It looks like we have some more time for another question. How long can the spheroid models be maintained in culture to monitor tumor-liver interaction?
So for the first, the 3D spherical culture condition, the suspension culture condition, we found that tumor cells, they have no problem, tumor spheroids, but the liver spheroids, indeed, especially hepatocytes, they gradually de-differentiate, and then so up to 7 to 10 days, then we would notice that those hepatocytes, even without tumors attack, they don't look like hepatocytes that much anymore. So that's why we gradually move on to this we call 2.5D culture condition. So we have normal cells plate on the bottom and then tumor spheroids on the top. For that condition, especially if we're focused on non-hepatocyte microenvironment cells, those cells can be maintained at 2D condition very well. So then for this 2.5D culture, then we actually have some condition up to 6 weeks. So, and then the interaction is still ongoing. Although we need to double check how well as it models the in vivo condition. But yeah, so the 3D hepatocytes, there are limitations, but for the non-parenchymal cells, they can be maintained actually for quite long, up to, at this moment, we know 6 weeks, no problem.
Thank you, Liqin. It looks like we have some more time for another question. What might have caused PMFs and IMFs to function so distinctly in your models, considering they were identical cells to begin with?
Yeah, thanks for this question. It's a great question. So that's actually what we are working on at this moment. So we can easily do single cell. We are actually doing not easily, but we're doing the single cell sequencing of these different cell types that mixing in our co-culture and see how their gene expression change over time, and how these gene expression changes associate with the cellular change that we are already seeing in the co-culture conditions. So, but just from the direct viewpoint, we noticed that the organization or the team up of these cells are different. So PMFs, they tend to form this fiber strands that they form very tight bonds between them already. So, but for IMFs, these tend to seem to be like more disturbed and then more disseminating and just mix not one by, but just mix within the tumor mass. So we think, for PMF and IMFs working differently, they also actually because of their neighborhood, their friends or their team members. So if there's a very strong team member coming together, then they can fight their cell, other cell types more strongly. But then if they're disseminated, then by themselves, then they tend to probably coordinate with the other cell types around them, try to survive themselves. So that's just some hunch that we get from observing the cell behavior changes there. But then we're again doing the single cell based analysis to try to pinpoint the molecular mechanisms, molecular changes associated with these functional changes.
All right, thank you. It looks like we have some more time for one last question, which is, what are the limitations of your models?
Yeah, the models are the in vitro models. So I guess that, it's just, the spheroids can never really model the cellular architecture that we see, in vivo. So we can add, the good things that we can add different cell types there, but, for how they organize together, how they form cell organization, there's just, there's a lot of things that we cannot control. And, one thing I like to point out is we cannot model endothelial cells in this model. So endothelial cells seem to be very difficult to be isolated from the liver. And of course, even you are able to get them efficiently, and then they don't form blood vessels like what we have in vivo. So and endothelial cells have been shown to be very, very important for tumor genesis, for obvious reasons that they provide all these nutrients, oxygen to the tumor mass. So that's one thing that is really limiting in this model. But we believe that if we continue optimizing our cultural conditions and then know more about how these cells interact, organize, then we can modify our from our end just to give them best condition for them to organize and interact just as close as to in vivo. But we're still working on that, and then hopefully one day we can pretty much tease out every single cell type's contribution to liver tumor genesis. But that's a long-term goal.
Thank you so much, Liqin. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Liqin, and we'll be back soon with the next talk.
Thank you very much.